Hello, good morning or good afternoon, everybody, wherever you are. Probably there is no good evenings yet. Maybe for Manila people, there will be good evening very soon. But by that time, hopefully, we will finish this webinar. Just to start, I'm Devendra Saroj. I'm based at University of Surrey, and my position is head of e- Environmental Health and Engineering Center, which is a WHO collaboration center for uh, human health and water quality. Um, I'm moderating this session jointly with Dr. N.B. Majumdar, who is here and who is the honorary chairperson of the International Academy of Environmental Sanitation and Public Health, IAESPH, who is hosting primarily this webinar for different people from different countries. I will very quickly go through the program that we are going through today so that it's clear to everybody what is the time commitment and what sequence we are going to follow. So this is the webinar program. So starting 10 a.m. UK time or 2:30 p.m. India time. Dr. Majumdar and myself, we are just introducing about webinar for the time being. We will not be going through the technical details of the topic, but we have got 4 speakers after us. Professor Manish Kumar from IIT Gandhinagar, India, Professor Gerdian Medema from KWR, Netherlands, Professor Davy Jones from the University of Bangor, UK, and then we have got Professor Arunabha Majumdar, formerly from Jadavpur University as independent public health expert in India. After that, we will have an open session approximately just after 11 a.m. and will run just after until 10:30 a.m. And this will be an opportunity for all the participants to basically participate in discussions, suggestions, and if any questions to speakers, they will be here to respond to. And we also have colleagues from Asian Development Bank attending today from Manila, and some of them will be present here and they will comment on and whatever they have observed during the session, so that it will help us formulate sort of action plan to look into what we can do to address the issue that we are looking at, which is basically focusing on very 2 specific things, but it may go beyond that. And those 2 specific aspects are wastewater surveillance for SARS-CoV-2, particularly as indicator for different things, as specialists are going to talk about, but also from broader perspective of wastewater-based epidemiology, looking into the COVID-19 pandemic, also learning from historical context and perhaps thinking on how this type of epidemiology can help many types of stakeholders in planning post-pandemic world or during the pandemic process. What are the suitable actions for making this world better? Without further ado, I would request Professor Dr. N.B. Majumdar to welcome everybody from IAESPH point of view, who is the host. And after that, we will go to Professor Manish Kumar from IIT Gandhinagar.
Thank you, Dr. Devendra. Ladies and gentlemen, very good morning for friends from the US and Europe, and very good afternoon for friends from India, Dubai, Qatar, Philippines, and Japan. I'm so happy to welcome you for this very interesting webinar entitled UK-India webinar on COVID-19 and wastewater. The topic would be sewage surveillance for early warning. Now, I'm thankful to our board and both the co-hosts for the support and cooperation. I'm thankful to Dr. Hannah Simcoe Reed, whose enthusiasm was the starting point for planning this webinar. I'm grateful to each speaker who have agreed to spare their valuable time to enlighten us with their research findings and their knowledge, and also long experience in handling the science and engineering of wastewater. COVID-19 pandemic has multiple implications. I don't need to mention them actually. Testing the maximum number of population is crucial for planning adequate management of this disease. You all know about that. Now, some examples as of yesterday, the figures. Now for US, 154,213 per million. UK, 202,677 tests per million. Singapore, 199,904. Bangladesh, 6,548. And India, 10,918. So much tests per million. So you can see the whole range. Now, this shows that countries with large population and high population density face a huge challenge. Let's take the example of India. For we have reached this above figure with a lot of effort and over several months. Now, for keeping a tab on the numbers, actual testing is the accepted method. Now, however, when there is a need for regular tests at different intervals of time to keep a tab on recurrence, it becomes extremely difficult. A new way has been found out by the researchers in several countries. Such work, I'm sure, would lead to a new set of indicators which would be easier and more practicable. In India, surveillance of sewage was successfully used for a long time to track efficacy of polio vaccine campaign. That was quite some time back. Now, for developing countries like India, there is another important factor. I'd like to mention this also because for us, it is extremely important. Now, the sanitation workers, such as those cleaning sewer pipes, the plumbing workers, or those working in sewage treatment plants, they are often exposed to raw or semi-stabilized fecal matter, exposing them to disease and even death. Now, the same is true for septic tanks and fecal sludge management systems. Only, I think, in sidelines, see the two-pit Sulabh Shauchalaya system, which has a 2-year moratorium for stabilization, possibly that doesn't have this risk. But that's a matter of further debate and so on. Surveillance systems will alert these people and their departments so that they can save themselves. Now, the same surveillance system can be used for other epidemics and pandemics also. Now, I'll end with a recent example, which was reported in the Smithsonian Magazine of 21st July. COVID-19 infection was reported in the sewage of Yosemite National Park, California, after the park was reopened in early June. Earlier, no one, whether living or working in the park, were seen to be positive. So you can see, still it was circulating in the sewerage system. Now, friends, our objective is not just this webinar, as Professor Devendra also mentioned. Our aim is to initiate pilot projects with suitable partners in India and other developing countries so that we can take it forward. Thank you very much. Now, Professor, you would like to call the participant speakers, or I should do that? Whatever.
That's all right. I think we can directly give floor to Professor Manish Kumar because he's probably ready for his presentation. Yes. So Manish, if you are ready, please feel free to go ahead. I presume your slides will be shared. That's great. So please try to stick to 15 minutes presentation, Manish, if you can.
Definitely. So thank you very much, Devendra, for giving this opportunity. So what I know that the audience here is entirely different, and they have the certain type of the acquaintance of this, and Dr. Majumdar has already mentioned good figures and that how it is important. So, but being a first speaker, I would say a little bit on that, and then I would just share my experience so far with this surveillance. So March, we all were locked down, and then at that time, we wrote this paper about a chronicle of SARS-CoV-2 part 1, because at that time it was several things to be known. Then there I wrote about transmission and treatment. This is the paper that we got published in 2000 in the May, and I would like you to see that. What is the one point that I want to bring it here is that at that time I was talking about an individual, how you can know without, not because of the symptoms, but just by a simple blood test. So it says that increasing neutrophil to lymphocyte ratio may give early sign of COVID-19. So at that time, I was also trying to predict early sign of a person getting infected. But when it started with the several, it goes gradually, then I try to know, because I am an environmental engineer by practice, so and by training. So then we have tried to know that there are 3 loops. And there were this SARS-CoV-2 was not a new. Many of us, even if educated people, thinks that SARS-CoV just came from some like just invented or something like that. But of course, there is a primary, and there is a lot of a story about the genetic viruses. But the cycle was going on in the wild, which was interfered by the human. And once we have been interfered, then it comes to the environment for sure. So getting into that story to the next step is that many of us, and then there were a flood of papers. Some people don't like about these things, but the point is that whatever we can do is the knowledge. And gradually nothing had changed, if I would say, between the lockdown period in March and now. The only thing that had changed is that we have gained several knowledge on that. And that's why we are somehow ready to say that, okay, let's go out and we can deal with it. Now we are more prepared. This study, which is the published, become online in June, and they say that there is no evidence of CoV transmission through contaminated water. So far what I have shown is that there is if some person is infected, there is not only one way of excretion that virus will come into the water bodies. There will be, if they face wash their faces or spit or anything, then the viable one, because according to me or some studies that excreted, if a virus goes through the digestion and then goes through the excretion, at that time viability of the virus becomes very less. It is fine. But when it is the other way around, then the viability of the viruses is much more. So what is happening is that gradually we were gaining the knowledge over it. And now this one is that no evidence is true as per the publication at that time. But now the viability or infectivity are coming into a broader picture or the light. Another thing is that like Dr. Majumdar, even the number, fascinating number, that even if they so many tests, and many times we now see that 25,000 new cases has been found, or 12,000 has been found, but it is against the number of the tests performed. If we performed less number of the tests earlier than the number of the things, so we should say the percentage of the detection rather than the number of the absolute number of the new cases, in my sense. But even if we will give the percentage detection, it is a observed case, and which will be entirely different than the unobserved case, which is still hidden. So we cannot make sure that who will come to us and whom a testing, the squad will go and test it. So it is a kind of clinical iceberg model where the hidden case will remain for a very long time. Then I had, we also wrote this revision submitted yesterday night, that if the virus, given the size of the virus, I am not talking about alive or not alive and like that, or viable or not viable, but it has the tendency, or the loop is such that it will reach to everywhere. It will reach to the aquifer, even the groundwater. There were actually this paper, we were arguing that it may go to the groundwater, and that is the new thing. Then the already rivers and wastewater. So far the point is that if there is a virus generated and coming into the environment, it is likely to go into the environment. So individual surveillance and environmental surveillance has the two dimensions of one coin, perhaps. And this is a picture again under review prepared by us, is that where we say that the clinical case can only be ensured about 1%. 2 or 3 days before, there was an article which was saying that the people will be more affected within the household or within those things. So even if somebody tests me, maybe I cannot be assured of my family conditions. So that's why. But if somebody took the sample from the sewage water of my colony, and they say that there is no gene found, I can be assured that there is no infected person in my community. So this and also so that's why the environmental surveillance can have the broader implication rather than the individual testing. And because I have the little time, so I would not take much. But what I would want to again emphasize that wastewater surveillance is not a new thing, and it has been used for the norovirus, like Dr. Majumdar has also told. And the biggest advantage is can be shown in this diagram, where you can see that there is a red, the red line, which is the number of gene detected in the wastewater, and this is a black line, which is the number of patients admitted in the hospital. And what we can find is that the difference in the peak, the patient peak was observed after 2 weeks of the peak that was observed in the wastewater. That means that we will have this kind of a advantage of 14 days or 10 days to prepare, even if the adverse condition is following up, or even if, so in this lockdown, we are not sure that where we are heading, whether the conditions are deteriorating or improving. There is no information about it, and it cannot be obtained through the individual testing. But for that, environmental surveillance is the goal. Of course, many people can argue that, okay, the virus in the environment. Of course, we have published this paper in last year, September. We were not knowing that another viruses are coming up. I would not spend a lot of time here, but what I can do, I can tell you that there is a little bit seasonality effect also is there. And there is a lot of progresses are happening. And here what happened is that we found that in winter, there were viruses, but in summer, in the environment, it could not exist, or it was not there. It is the case of the norovirus. So I would not emphasize on here, but because I want to finish on the 15 minutes. So about coronavirus, again, coronavirus was not new. Kyle Beebe in 2013 in ESNT, they took the 10 sample, the 10 sewage sample, and they screened the sample with the several viruses, DNA viruses and RNA virus. And coronaviruses, 83% detection was of the coronavirus. So of course, SARS-CoV-2, the particular strain that is also of 6 types or 7 types. There are many, the research is going on in that field. And they said, they concluded that now the abundance of the respiratory viruses are getting higher in the human sphere. So that is the one aspect. And then the following up that, there was talked about that there is a expression. And this all makes it possible that we can do the environmental surveillance. And that is likely or meaningful to do that. And then there was a consortium made by the Kyle Beebe, also many of us are part of that. So, I mean, here there are all the speaker perhaps is part of this collaboration and effort. And anybody who would like to join even now, they can do it. So, because I would not give a lot of emphasis here, but there is a genome, like there are a SARS-CoV-2 will have the different genes. So what genes are being detected is also there is no similarity between among the globe. Around the globe, there is no similarity. Even in the India, some are concentrating on ORF, 1AB, S and N genes, somebody. So there is also E genes and somebody only N and E genes. So there is a little bit discrepancies or little bit dissimilarities between the results. That's I also wanted to just make a note. Then we did the, coming to our experience, we did the sampling and then the 2 days, and then it is at that the concentration step is very much important, that we have to do 500 times or 100,000 times concentrating the sample, because in the water genes are very diluted. So that is, but in this case, because lockdown was there, we tried to go with the polyethylene glycol and NaCl, so that we precipitate the virus and then analyze it. So sometimes there were some methodology that can be used for so in an Indian context, we have some kind of tweaking is possible. And recent study also showed that, and at that time we observed that people said we have to store the sample in minus 20 degree, or at least it started with the minus 80 degree. I didn't have a facility for minus 80 degree or minus 20 degree. So then, but many times we just put the AC on and we kept, and after 3 days we analyzed and we go. And now it has been confirmed, of course, not by the peer review, but of course the Kyle Beebe team has informed that they have seen that at 40 degrees centigrade, it can sustain. So those things were there. Of course, this one, a lot of you are already aware of it. But what I want to say is just a glimpse, a snapshot, that here there was a influent and effluent, and we have taken it, there is a gap of 20 days almost, and then there is a number of the patient was almost the 3.8 thousand becomes the 5.3 thousand as a number of patient, and copy increased by 7 times. So it is the number, it means, but if I would, you will ask me, can you give me a coefficient for that, number of people and number of the gene detected? No, this, but having said that, you can see that the number of the patient can lead to the increase of the number of copy. That is also one of the points you have, I want to be noted down. And another point is that there is nothing in the treated water. In the treated water, we could not find any gene detected. Okay, fine. So then what happened is that we have done this, the comparison, and comparison we have done with, till then there was only one, the few people who have done that work. And so then what is there is that, so we have the 100% detection and 0% detection in the treated water. But when we saw it closely, what we found that from France, it was reported that even in the treated water, they had 75% of positive detection result. Earlier, the other study has not even taken the sample of the treated water. We did take the treated water sample, but others have not taken. But there was then we have closely had a look on that. And then we found that in China, actually it is very opposite. In the before treatment, there was no gene detected, but after treatment, there is a gene detected. And then in Spain, there is a, I mean, the log reduction is just 0.1, around 0.1, or so even 1 log reduction is 10 times or then something like that. So it is very low reduction of that one. Now, so then what we did is that we took the sample from each step, and now this is under review in Journal of Hazardous Material, and we have found that many times the influent had no conclusivity. We could not detect a significant number, but gradually it had some number, and then the final water didn't have something. So it means it is not very simple, a state of a simple. But for Indian context, what is more important is this septic tank. The Chinese one that I have shown you is the before treatment not detected was the case when they sample septic tank treatment of hospital effluent, and which is the major case in India. And if it is a major case in India where we are doing septic tank treatment more, so this is a very alarming condition. We cannot, that before treatment there was nothing, and after treatment there was detected. So that is also the third point that I want to make here. And then so then what we did is that this is my second last slide. So so what we have done is that we have tried to find that infectivity, because it is very much like people say, but it is convenient to say. And I like that in your introduction of the flyer, it is written that infectivity has to be investigated further. And what we have found that this is under review in ESNT, this is that wastewater, how much copies were, even after the effluent, we found that 1 to 2 log of the SARS-CoV-2 will be present there, and which is likely to, even after the dilution and decay, is going to remain 2 log condition. So which means that whatever the disinfection we do, of course, there is a variation between membrane technology or UV on or A2, the different technologies. The reduction is different, but even if whatever we do after effluent, there is a likely to have some copies remain in the water bodies, and this is going to affect the our infectivity. So for 3 action, we have tried to determine the infectivity, and we saw that and we plotted one expected dose per event and chance of infection. And we have seen that there is a chance of infection, and it is very important to have a quantitative microbial risk assessment before we go through. So I would just finish with that. Of course, there is a different paper device and like that, because my time is up. What I would say is that in Indian context, even the several things has to be come into the picture about the wastewater, I mean the antiviral drugs handling and those things that also we have told. This is of course the last slide where this Ralph Hayden, according to him, that in Indian case, maybe we have to have the supplementing this. It is impractical actually, the individual testing. So environmental surveillance is must. This is certainly the last slide. After that, I will show you the thank you. These are the issues that I have enlisted, but I want my audience to think about only the last 4. Concept clarity that this is a supplementing method, not a replacement. So individual testing has to be there. Number of people infected or trained or screening. What we want to know is very much important. People are confused that we will know about the number of the people, which is still very far. And the distinguished fellow speaker will tell about that. And but the point is that in India, we have to have participation of a stockholder and willingness of the policymaker, and and which is still happening. But Professor Majumdar will be telling about that. And there, the capacity building is necessary, for which this webinar is one of the step I feel. So thank you very much, and yeah, thank you for giving me opportunity.
Thank you, Manish. It has been really very interesting presentation, especially to start with, because you know, you really summarized state-of-the-art situation in India, which we are all very, very much interested in, because probably that is the similar situation in many Asian countries and other developing countries. So I think you have raised a number of queries and questions that might be coming up, but we will address them when all speakers have finished, because there might be very common questions. Is that right, Professor Majumdar, Dr. Majumdar? Is that okay?
Yes, yes. So now maybe we can invite Professor Khatya Medema for his presentation.
Yeah. So Professor Khatyan Medema, he's based at KWR, Netherlands. Many of us have read his work a lot, also seen in media. So it's very good timing after the Indian talk to look at the research done in Netherlands and other relating to other European countries, perhaps. So Professor Medema, you have got about 15 minutes. If I'd be grateful if you can stick to that time. Thank you very much.
Thank you very much, and thank you for having me here. Do you hear me and see my slides?
That's perfect. Yeah.
Okay, super.
And we can hear you.
Thank you. Yes, I will talk about the sewage surveillance and from the Dutch and European perspective. So I'll go through the slides quickly. First, why do we do surveillance? And that is to look at trends in all kinds of diseases, and in this case, particularly COVID-19 trends. And we are in Europe, we are in most countries in the situation where we are past the first wave, and we are all anxious to see if a second wave is emerging. And surveillance and also sewage surveillance can help there. Sewage surveillance, I think Professor Kumar also showed a pyramid. I think we all know that there's the infectious disease pyramid where you have the cloud of people in society, and a part of those are confirmed cases, but many people carry the virus but are not captured by the regular testing, even though testing has been increasing. But if you just compare antibody results versus test results, and you see a factor of 10 or 20 of people are not diagnosed while they have been exposed to the virus and have carried the virus and produce antibody response. So there, sewage surveillance is in our country now on the national dashboard of wastewater. So wastewater is a population surveillance. I think you're all aware of that, and we take one sample where you monitor 100,000 people or 1 million people. But then the question we had at the beginning is if it is sensitive enough and if it's quick enough. The basic idea, and I think Professor Kumar also already showed the overall method: concentration of and purification of the SARS-CoV from sewage, extraction of its RNA, and then using RT-qPCR against, well, we've been using these targets, but as Professor Kumar was saying, there are many other targets that have also been employed. The basic concept is then, of course, that you see if there's more COVID-19 patients in the population, you see more SARS-CoV-2 RNA in wastewater. So that's what we, when we started out in early February, when there were no SARS-CoV-2 cases in our country and no SARS-CoV-2 RNA in the wastewater, we wanted to see if we saw something, and that's what we've been publishing on. So early March, 1 week after the first case arrived or was reported in the Netherlands, we saw the virus RNA in wastewater of 6 cities. And in one of those cities, that was about a week before the first COVID-19 case was reported in that city. And we saw the SARS-CoV-2 RNA concentration increase with the people increase in COVID-19 circulation in those cities. And then, of course, we questioned, was there a correlation between the two? And here you see the correlations we had between cumulative cases per 100,000 and gene copies per ml of wastewater. And we've published that. So we feel that at this early stage of the pandemic, wastewater was a reliable signal when we look at the circulation of the virus in the population, and it also was an early signal. So as I indicated, it was in one city 6 days before the first reported case. We later saw that again in another city. Of course, then the question was what happens next, and when social distancing measures and lockdown measures were implemented. Well, we saw here's the results of Amsterdam, reported cases, reported hospitalizations in gray, and reported N2 gene concentration in wastewater in orange. So you see there's a nice match between what we see above ground and what we see underground. And also that there was an early signal, and now of course we are at the end of the first wave, and the question becomes, what do we see next, and when is a signal that we see in wastewater signaling a second wave coming up? So to show you the results of other cities that we have, these are similar results in each of the cities, and you see in the bottom left, that we saw a small second wave, at least in the wastewater, but not so much. Although if you look carefully, there could be a rise, but there was also a rise in testing. And as Professor Kumar rightly said, we should also consider the number of tests that have been employed. Okay, inspired by results that we had and many others, and I think Dave Jones from Bangor University also, will present results of a similar nature. The European Union is now stepping in, although Europe does not have a health mandate. That's all in the member states and not at the Commission level, but they can do supportive actions, and one of the actions they are employing is to do a EU-wide wastewater snapshot. And we do that with the partners you see listed there. Joint Research Center of the European Commission is the key partner there. So the use case of this umbrella study is that to demonstrate to the European member states the feasibility and the value of wastewater surveillance. It comes, the other element is that it aims to connect all the national and regional and local initiatives that are ongoing in different European countries. And so far, I cannot disclose the results, unfortunately, in more detail, but 52 cities tested, 28 were positive, and this is the range of concentrations that you see. And there seems to be a match with countries where there's a lot of virus circulation with high concentrations and the other way around. So I'll talk a bit about use cases. This is in the Netherlands. You see it's in Dutch, but you recognize the dashboard on coronavirus. And you see one of the elements of the dashboard in the Netherlands is sewage surveillance. And you see the line of going down over time. So there's a national average of the 29 sites that are monitored. But and so it is the general trend, but of course the more value is when you go down to regions or cities to see if there's areas where there's a resurfacing of the virus. So it could serve as an early warning and then support decision on increased testing in certain areas, social distancing in regions, what we see now happening in different places in Europe. An overview, not complete, but to give you at least an impression of what's happening in Europe. There are many countries embarking on wastewater surveillance in larger national studies. That's more on the top, although you'll hear from UK, there also are national studies there. You see more and more that it is adopted as a system for surveillance. Still in the early days, and still there is it's environment and health sector that have to come together on this, but we are clearly moving in that direction. Well, this is showing again Amsterdam. Why I want to do that is that I think particularly on this level we can make the relation between surveillance data in sewers and surveillance data in people testing, and also look at a circulation of virus genotypes and also what we know from antibody testing. So I think these focal points are also points where we can bring the surveillance systems together and calibrate them against each other if you wish. Another example from Valencia in Spain, an example where we are going down into a city, one of our larger cities, where we also have the objective to look at distribution in different areas of the cities with different ethnic groups and different socio-economic conditions, because we believe there are biases in people testing, and sewers can be an objective judge of that. Hospitals have been, or hospital monitoring has been suggested, nursing homes, school campuses, all to look at circulation of trends, or virus circulation trends in the population inhabiting that. And for schools, particularly also silent circulation among young people that are becoming more in the attention. Cruise ships, airplanes, just suggested in Australia, where you could monitor entry of the SARS-CoV-2 in a region. And last, the events to look at if there's circulation of SARS-CoV-2 and understanding the role of the events in the spreading of the disease. So I'll end with what I see as a scale up of this environmental surveillance and the growing pains there. We see in Europe, and I see it also in other regions, that because it's on the border between environment and health, there's always the question who is the responsible party. And I guess it's in this situation, it's the two worlds that have to come together and make up their minds about who is going to issue payments for sewage surveillance. If you really want to scale up, it's also quite an endeavor. We see in Europe, it's national institutes or consortia of institutes and laboratories and utilities that can do the job, but it does require a lot of collaboration. And then there's questions of scale. Do we do all the population like in the Netherlands, but we're a small country, or do we do sentinel cities? And at what frequency should we test? How to ensure data quality, and how do we translate these data to particularly decisions to control COVID-19? So when do we call a rise or a level in wastewater critical, and how does the RNA signal in wastewater relate to the circulation in the community? So with that, I thank you for your attention.
Thank you, Professor Khati and Madema. It has been very useful addition to what Professor Manish Kumar just gave in his presentation, and gives a much broader picture. It's very useful. Again, we will keep the questions and answer sessions together after two more speakers have presented their work. Next in list is Professor David Jones from Bangor University, UK. And we can go straight to his presentation. Is that all right, Dr. Majumdar?
Sure. So welcome, Professor Davy Jones.
Okay, great. Can you see the screen?
Yes, we can.
Okay, brilliant. Thank you. Yeah, thank you for the invitation to present. It's a great honour to be here and to present some of the latest data from the UK. And thanks to the Professor Kumar and Madema for doing a fantastic job of setting the scene. I'm based at Bangor University, and jointly I have a position with the University of Western Australia, although since COVID-19 I've not been able to undertake my duties there, been confined to Wales. So we are funded through the national government, through what we call UKRI, the research councils, and through the water companies. And also we linked up now with Public Health Wales and Public Health England to be able to use this data effectively. So just thanks to those, really. So, I mean, we've heard about this already, so I'm not going to spend too long talking about it. The wastewater is essentially the waste products, the dregs of society. And, some people say that the wastewater never lies. It tells you everything that's going on in society. So we're trying to get this snapshot of public health through analysing wastewater, and particularly pick up pre-symptomatic or asymptomatic cases. It's been successfully used for polio and various other things, and there's good data for norovirus, etc. The key question is, as said in the last talk, really, does this really work for SARS-CoV-2? Can we use it as part of our national surveillance? So just to give you a bit of a background of where we're coming from, you can see here the numbers of viruses that are shed in feces versus those that are also shed from respiratory droplets. I have no doubt that SARS-CoV-2 is primarily a respiratory droplet spread disease, but you can find it in high abundance in fecal material in infected people. And that is the same with most respiratory viruses. It passes through the gastrointestinal tract. So the basic principles that we've been studying really are this: monitoring the amount of SARS-CoV-2 in the community, monitoring it in the wastewater treatment plant, monitoring it in effluent, and then looking at potential impacts on coastal ecosystems and fresh waters, and basically doing a risk assessment of that. In the UK, we've had a lot of research on this in particular, not on SARS-CoV-2, but in previous years about accumulation of pathogenic viruses in the environment, particularly in food, and the link between human wastewater being heavily contaminated with norovirus, and then people consuming raw products. In this case, it's oysters or shellfish, and then causing outbreaks. And that's hepatitis, norovirus, lots of things like this. So the UK government has invested quite heavily in looking at certain viruses. So we have a track record in looking at surveillance for norovirus and other things in particular. And this is basically led to the development of a whole range of methods which allow you to concentrate viruses from water samples from the environment and from wastewater. In this case, we're looking at the recovery of norovirus, human adenovirus, sapovirus, mengovirus as a control. And what you can see is that you can develop these methods. And one of the really interesting things is that the one method is suitable for norovirus is not so good for sapovirus and vice versa. So we're still trying to find the golden method to identify, reliably extract viruses from samples in a quantitative way that recovers them all. And that is actually a really tricky thing to do. We've tended to concentrate on using ultrafiltration combined with PEG precipitation. And some of the work that we've been doing for norovirus, for example, which shows that it's an effective tool. Here we have the graph which shows genome copies per litre on the y-axis for wastewater sites, river sites, which is water, fresh waters, sediments, and for shellfish, so in marine waters. And what you can see is that there's high concentrations of viruses in the wastewater, and gradually, as you would expect, they become diluted, but they do accumulate in shellfish, and which then we enter the human food chain. Norovirus shows seasonal patterns. Other viruses like adenovirus don't, so they're good markers. And what's critical here is that in the environment, these viruses can accumulate in high concentrations, which poses a risk considering that the infectious dose for these viruses is 10 to the 50 viral particles. Now, we don't know what the infectious dose is for SARS-CoV-2, but we expect it to be in the region of 100 to 500 viral particles. We've also been doing metaviromes for viral surveillance, which basically led to this particular project, looking at all the different viruses that you can see on the left-hand side there, lots and lots of different taxonomy groups, of course. And what we've been doing is using that to pick up wastewater at the bottom, WW, in foods, in rivers and in sediments. You can see that in certain parts of the environment, these viruses are accumulating. And sapovirus and norovirus or rotaviruses is another good example of viruses that we can pick up from the environment. So all of the tools and techniques are there to be able to do environmental surveillance of SARS-CoV-2 and other viruses. And I think, you know, as we become better at sequencing technologies, etc., we're becoming much more used to using these approaches to basically evaluate risk. So how far have we got in the UK in SARS-CoV-2? Well, we know that, you know, not everybody who contracts the disease will be shedding. Possibly about, it looks like about 1 to 3% of people infected will be releasing in urine, and about 35% of people will be releasing it in fecal material. There seems to be no great correlation between fecal shedding and diarrhea, which is one of the classic gastrointestinal symptoms that people are using. That's current evidence from the UK. So the gastrointestinal symptoms, which you can see on the bottom here in the yellow bars, suggest that these are only in a small number of clinical cases and clearly not present in asymptomatic or pre-symptomatic cases. So we can't really use, our view is that we can't really use those numbers as an estimate for fecal shedding rates. But in terms of the quantifying the amount that is in the samples, we know in comparison to respiratory fluids, which are 10 to the 5 to the 10 to the 11 genome copies per mil, the amount in fecal material is about 10 to the 2, 10 to the 7 genome copies per mil, so a lot less. So the signal is much more dilute, and that's why all of the conclusions are that respiratory droplet transfer is the primary mechanism for SARS-CoV-2 transmission. We're not discounting fecal oral completely, but all the evidence suggests that it's not important in comparison to the main event. If you look at release rates, on average, a person who's infected is releasing about 10 trillion SARS-CoV-2 viral particles per day. And then when you look at the amount in sewage, this is about 10 to about 10 million or 100 million, depending on the level of infection of the individual. Evidence suggests that pre-symptomatic and asymptomatic people, at least in this country, are not shedding at higher levels. And the limited quantification for hours, once we've taken into account all the uncertainties, means that we are detecting one case in the community, so one case in 10,000 people. So it's quite good for most of our urban centres. We recently wrote this review, and it's currently in the process of being published. It's been accepted by the journal. What it says is that, based, at least for the UK systems, fecal oil is not an important transmission pathway based on the sewage systems that we have. That catching SARS-CoV-2 from beaches, recreational waters and drinking water is minimal risk. It's completely different to norovirus, which is much more hardy in the environment and there in much higher concentrations. And that public toilets are high-risk environments, but that's not through the fecal oral route. That's predominantly through respiratory droplets being in those environments. So that should be out shortly. So we've been screening these large urban wastewater treatment plants. Here we've got Cardiff and Wrexham, two of our big urban centres. They are treating wastewater from about 900,000 people in certain cases, down to about 100,000 people. So that's the sort of levels that we're dealing with, close to 1 million. We're collecting the wastewater, doing the classic centrifugation. We used what's now known as the Medema protocol. It'd be nice to have a protocol named after yourself. And I think it's great. We've been using that. We've now switched completely to the PEG precipitation method rather than the ultrafiltration method. We've shown that they both give comparable results, and it's much cheaper. We've gone through to an automated RNA extraction procedure, and we're currently using qPCR, but are switching over to digital PCR in the coming months. So just looking at some of the data from the UK, we're using the N1 gene as the target. We have used the E gene, but we're focusing on N1 because N1 works really well for us. We're using pairs, which is an envelope control as envelope virus as a process control. So adding that in and looking at its recovery so that we can correct for that if necessary. And we're using crassphage as a human marker. So that allows us to basically ratio the amount of SARS-CoV-2 in the samples versus the crassphage human marker, which is always released in human feces. So it means that we can we don't need to account for flow using chemical markers and things like that. So, and crassphage is there in high abundance, and we've tested that one before quite extensively. So what you can see here is that our lab was closed due to COVID cases, actually during the peak of the epidemic. But when we went into lockdown, we reopened the lab a week into lockdown. So when they sent all the people home, and what we can see is really high concentrations, or relatively high concentrations, at the start of the lockdown period. And then we've basically rapidly declined as the number of cases basically maps exactly the cases as Prof. Medema's had. But in certain cases, we've had these outbreaks. This one here was a localized outbreak associated with meat processing plants in northeast Wales. And this is now, which actually occurred, the clinical cases were happening 2 weeks after that. So it's a pretty good indication, at least from this perspective, that we do have some advance warning if you can process the samples fast enough to ensure that you have that advance warning stage in there. So we've still got samples backed up from January, still waiting for testing to see if SARS-CoV-2 was circulating in the population before the first clinical cases. I don't think that will be the case. But generally speaking, the lockdown measures in the UK have been incredibly effective. So the findings so far is that we've repurposed all of our classic capsid assays to the recovery of envelope viruses. SARS-CoV-2 is clearly detectable when the levels are high in the population and responds to that. The levels in effluent are really low. There's no risk to sewage workers, at least under the current sewage conditions that we operate in the UK with PPE, etc. All the evidence suggests that actually the sewage workers have been less sick during the COVID-19 outbreak. than previously. So no evidence that there's any risk of transmission from sewage to workers either in the sewage network or at the plants themselves. We still can't reliably estimate the number of infections in the community because we don't understand enough about fecal shedding. Although people are doing this, I think it's actually highly unreliable. In the UK, we've been asked many times whether we can do care homes and schools, but there's tens of thousands of these, and it's just impossible to do it on a big scale. But it is suitable for prisons and other high-density environments, airports, and industrial areas which are high risk, so where we have meat processing plants and people that are in close contact with each other. So can it be used as an early warning system? The evidence that we have suggests that it can, if you can process the samples fast enough, but there is still a lot of variability in the data which needs to be sorted out. We are worried in the UK about the impact on in the environment. For example, we know that raw sewage, this is a recent newspaper headline, was discharged into rivers 200,000 times. So we have a lot of raw sewage going into our river system, and the actual public health messages that are going out at the moment are suggesting that people are putting signs up along the rivers saying don't swim in the water because it contains SARS-CoV-2 and you'll catch COVID-19. Now this is highly inflammatory. We don't believe there's any evidence for this, but we have noted that the study from Turkey picked up infectious SARS-CoV-2 from wastewater influent, and I think from effluent, in a very, very small number of cases, but they have incredibly high numbers in their sewage. Well, so what we've been doing in the last sort of couple of weeks is essentially modeling viral plumes of SARS-CoV-2. So the little graph at the bottom shows the rate of infections in the community and a theoretical discharge of sewage off the coast. So there's a sewage pipe that comes in off the coast here, coastal discharge, and the red to the blue tells you the amount of virus that's circulating in that water. Clearly this is just the RNA signal because we don't know whether it's infectious, but it gives us an estimate. And from this type of approach, we can see these are high-risk areas because they're tourist beaches, they're also shell fisheries and recreational waters. And that's the sort of work that we're doing now, is to quantify this risk, to actually look at the levels of abundance that we're getting in water. So in terms of the UK as a whole, we're currently operating 2 plants at Wrexham and Cardiff, wastewater treatment plants, where we're doing hourly sampling for a 3-week period to quantify uncertainties, which is phase 2. Phase 1 is the data I just presented, and phase 3 is where we're going to sample 80 sites across the UK. Some of them are already underway, some of them are on planned. There's a focus on urban centres. We're monitoring them 3 to 4 times a week. We're moving towards a semi-automated quantification system and to digital PCR. And as Prof. Medina had, we're linking through to the public health portals. That's just about to go live. And we're also measuring other viruses of interest. So for example, antivirus D68, because then ultimately we want to test this now for future outbreaks. And sequencing has been done, just needs the data to be analyzed. And we're looking at infectivity in sewage, not in our group, but in other groups that are associated with us, which will basically answer that question about whether there is a risk or not. So that's all I have to say. I hope it was useful for people, and thanks very much for listening.
Thank you very much, Professor Davy Jones. It has been yet another good addition to previous 2 talks, really. And I'm sure that there are people having many queries about some of the points you raised. So to participants, I can request that if you already have some questions in your mind, you can type them into Q&A chat box that you see at the bottom of the window you're looking at, and we can pick up later those. But equally, if you wish to write later on, that's also fine. I think the next speaker, who is the last speaker before we go to discussion, He's Professor Arunabha Majumdar. He used to formerly work at Jadavpur University, an expert of public health in India. And we wanted this final presentation because it's very important from public health point of view to understand history, present, and potentially future, and how it is relevant to public health bodies and different stakeholders. So Professor Majumdar, you can start whenever you're ready and your slides are up. And
Right.
please limit your talk
Yes,
to 15 minutes if that's possible.
yes,
Thank
yes.
you.
Thank you, Dr. N.B. Majumdar, Professor Devendra, and respected speakers already spoken on this particular subject. COVID-19 is everywhere now. People are suffering. The government are taking different measures to curb this particular disease, which is very much contagious. Now, the testing in many countries is limited. Initially, Dr. N.B. Majumdar mentioned per million testing is different in different countries. And in India, where we have 135 crores of population, our really the testing is limited. So therefore, we have to look into whether we can have the wastewater-based epidemiology, the study on sewage, analysis of coronavirus in sewage could help to the health personnel, health department. Next slide. Next, please. Yeah. Now, if we look into early in the COVID-19 pandemic, research revealed that people infected with virus shed, and this shed viral materials in their feces. That insight prompted an interest on sewage epidemiology. That was studied earlier. Even in 2005 in China, the study was made. By sampling sewage and wastewater at different parts of the sewage network, one can gradually narrow an outbreak down to smaller geographical area. This is the advantage because we have sewerage network. We have main line, branches, laterals. So therefore, we can zero down to find out exactly which area the infected people are residing, and then enabling public health officials to quickly target intervention in those areas which are at great risk of spreading the infection. The viral RNA, as mentioned by earlier speaker, can be extracted and subject to RT-PCR analysis for detection of viral genes in collected sewage samples, and this has been elaborately mentioned. Next, please. There are problems to be solved in order to maximize the accuracy and value of sewage-based surveillance system. Already different speakers have mentioned regarding this, and they also emphasize on this particular issue of maximizing the accuracy and the value. Related with respect to the infection, how this could be related with respect to assessing or predicting the future infection that may come. This is a question that needs to be considered. Also, there are very many issues relating to the sampling, preservation of the samples, the transportation, and the testing, accuracy of the testing. The propensity of the virus to break up when it is in water, the effect on the other contaminants, and how many sampling points need to be included in the city and area network in order to build up an useful picture of the outbreak are the challenges to be considered. So therefore, this is also need to be looked into, that other contaminants, because this sewage and municipal sewage often mixed with the industrial wastewater. In India, there are very many instances that both the wastewaters are conveyed through sewer or open drains. Given the shortage of COVID-19 testing, it is hard for the health authority to assess how widespread the disease truly is, and researchers are therefore using the proxy. I consider this work is a proxy, measuring level of coronavirus in untreated sewage to see how much the infection is circulating among the people. Next. Now, if we look into 2005 China study, the study was conducted to assess concentration and detection of SARS coronavirus in sewage from hospital by Wang. Their attention was focused on possible transmission by the sewage system because laboratory studies shows that patient excreted coronavirus RNA in their stool. Okay, next. Yes, the study also said to explore whether the stools of SARS patients or the sewage containing the stool of patient would transmit SARS coronavirus or not. And the researcher used a style of electropositive filter media particle to concentrate the SARS coronavirus from sewage of hospitals receiving SARS patients, as well as cell semi-nested RT-PCR and sequencing of genes to detect and identify the virus from the sewage. This was done initially, and they were successful in looking into this particular genes. Next. The said study researcher found that virus can survive for 14 days in sewage at 4 degrees centigrade. This needs to be validated now with respect to different studies that have been carried out in different parts of Europe, already mentioned by other speakers. So what is the duration it can survive? This study says that at 4 degrees, 14 days, but at 2 degrees, 2 days, it is remaining at 20 degrees centigrade. Now, when we look into the Indian scenario, our temperature at the present moment is varying between 22 to 26 degrees centigrade, sewage. So therefore, it says that 2 days at 20 degrees centigrade. Now, this needs to be validated, and its RNA could be detected though the virus had been inactivated. Now, these are also looked into because now we are getting different type of studies and different results are coming. So definitely the new studies will give a path or pave the way for future studies. Next, please. This has been mentioned by Dr. N.B. Mojumdar also. Poliomyelitis eradication was a challenge to the whole world. And polio vaccine need to be given to the babies to control the eradicate the disease. And you know, in 2014, India became polio free. And India did a wonderful job in this scenario. Now, Wastewater is getting monitored for polio virus. So therefore, this definitely is an indication that the coronavirus could be an indication if found properly in assessing this particular disease among the people. Next, please. Paris study recently, a lot of studies have been mentioned by different speakers, but this particular study I like to mention, recently sampling sewage across Greater Paris, reported in 21st April 2020, for more than 1 month, researchers have detected a rise and fall in novel coronavirus concentration that correspond to the shape of the COVID-19 outbreak in the region. So this particular study, rise and fall, as mentioned by other speaker also, they have found where a lockdown was imposed for suppressing the spread of the disease. Higher concentration of virus in wastewater or sewage correspond to higher number of infected people who contribute to the sewage system. And this has already been mentioned by the earlier speaker, especially by Professor Darve Jones in his presentation. Next, please. Scientists in Israel found SARS coronavirus RNA in sewage sample in STP in Tel Aviv. KIWA Water Research Institute, we have heard from the professor from there, Netherlands carried out testing of flow-dependent 24-hourly composite samples. So this is a question to be decided whether we have 24-hourly composite sample. KWR Water Research Institute running composite sample for 24 hours, whereas the testing that was conducted in Queensland, Australia, by cutting composite sample prepared from grab sample taken during peak hours 8 to 10 a.m. So therefore, we have to make the protocol according to this type of studies. And in India, accordingly, we are developing the protocol, and I can little bit discuss on that next. Next, please. Scientists from IIT Gandhinagar and Professor Manish Kumar already mentioned his studies in collaboration with Gujarat Biotechnology Research Center, Gujarat Pollution Control Board, and they found in Ahmedabad the SARS-CoV coronavirus. And his studies we have already. A group of researchers of B. Lal Institute of Biotechnology, Jaipur, Rajasthan, in India, has shown the presence of this viral genome in untreated wastewater samples from municipal sewage treatment plant and hospital in May-June 2020 in India. Chennai Metro Water, they detected using RT-PCR technique presence of coronavirus RNA in sewage sample collected from Chennai city in India as part of preliminary study, April 2020. Next, please. Now, if we look into the wastewater management, our mega and metro cities, we have a lot of mega metro cities. These are mostly covered by the sewerage network. But there are some places in this mega metro cities which are yet to be covered by the sewerage. Most of our cities and towns are unsewered. We have more than 7,700 cities and towns in India, and most of these cities and towns are unsewered. So we have on-site sanitation system. So on-site sanitation system are practiced in unsewered areas where we will find septic tank mostly. But where we have sewerage system, we have off-site sanitation system and sewage treatment plant. But in rural areas of our country, we have on-site sanitation system, and more than 65% of our sewage in India is discharged without treatment. We are taking up now measures for more treatment. Tank is used mostly as an on-site sanitation system in cities, and in rural areas, twin pit port flush toilet is used mostly by the rural population. The advantage of twin pit port flush toilet is that the sludge is getting digested. It is not like septic tank. As per existing rules, liquid waste of hospitals are to be treated in effluent treatment plant. So in many we find that our hospital waste are getting treated in effluent treatment plant. In order to control water pollution in rivers, schemes addressing we are addressing interception, diversion, and sewage treatment, and that has been taken up for many cities and towns for abatement of river pollution. Next one, please. Now, sampling is very important. Study, if carried out through real-time monitoring basis, it can be sewage salis sample. It can predict presence of coronavirus infected patient showing symptom as well as in a locality, ward, zone, slum, gated community, city or town. This study can predict the possibility of second wave of outbreak as mentioned by other speaker of COVID-19. Of COVID-19 outbreak can be assessed by extensive sewage and salis sampling and testing, but the location of sampling point is considered to be very important. As we have heard also, it must be selected considering the existing sanitation system of the area. Now, sampling can be grabbed, can be composite, and duration and interval of sampling is very important, that need to be decided. Now, we suggest that it may be composite by collecting grab sample during peak hours. Our peak hours is between 7 a.m. to 11 a.m. So this can be the composite site collection by collecting grab sample and then mixing. Now, interval of sampling may be alternate, thrice a week, or it can be twice a week, depending upon this testing infrastructure. More we test, it is better. Okay, next. Next. Now, samples to be analyzed, this is suggestive as per our developing protocol. This tool of COVID-19 patient, then sewage sample from COVID-19 hospital where sewerage is existing. Then sewage samples from septic tank of COVID-19 hospital where underground sewerage does not exist. In cities, sewerage network may be divided in small zones and sewage sample may be collected from exit point of each zone. That will be the advantage. then sewage sample from quarantine centers, sewage sample from isolation centers. So we have different quarantine centers, isolation centers in cities and towns. Now, next. Raw sewage sample, which is influent entering into the sewage treatment plant. So sewage treatment plant influent as well as treated sewage sample, as also discussed by other speakers. In unsewered towns, sewage sample from septic tank. So it can be from hospital, quarantine center, isolation center. Sewage samples from surface drains are to be collected for testing. And in villages, sewage samples are to be collected from on-site sanitation system, 2 feet port per standard. But we are recommending a community-based sanitation system for the collection and testing, not individual. Next, please. So therefore, finally, concluding, we can say that if sewage sample analysis for RNA coronavirus is carried out regularly, then it could become an early warning system for future outbreak of the disease. And that has also been highlighted by different speakers earlier. Now, viral particles in sewage sample is the collection of virals shared by many people in the community and is akin to a pool testing. So therefore, this is basically a pool testing. And if it is identified, we can have the area, mapped area, and how many people are residing there can be understood. There is no other alternative but to test the swab sample by RT-PCR. This is must. Sewage sampling not a replacement to the testing for infection in people, but only a supplement. So therefore, this is a supplement we do consider, and this will be helping the health department, the public health department, for tackling this particular disease. Interference of other materials, microorganisms, if any, during the laboratory testing need identification and rectification. So this is one challenge we do consider, and the microbiologists, virologists are looking to this particular aspect, and the result will be definitely helpful for us. Next, please. Interpretation of sewage sampling testing results in predicting extent and magnitude of the disease COVID-19 in the community will be a challenge. This has been already discussed by my friend, Professor Jones. This is actually challenging one we do consider, this interpretation. However, considering a high percentage of asymptotic and undiagnosed cases, and this could be 70–80%, asymptotic people are there, persons are there, they are undiagnosed. Sewage epidemiology is in the absence of adequate clinical surveillance can provide a critical information required for the management of public health crisis. And herd immunity, when we look into, certain studies are being carried out, different countries, even in India, the studies have been carried out, and it has been found that the herd immunity is on the rise. In some cities and towns, it is ranging today between 20 to 26%. We are expecting this herd immunity to achieve 60%. Therefore, this particular issue of herd immunity is also very important in this particular thing. So thank you for your patient hearing. Namaskar. Next, please. Thank you.
Thank you very much, Professor Majumdar, for summarizing and presenting different aspects, particularly considering some developing countries, specific aspects that we wanted to hear as well. So now we have had very different kind of topics covered by our 4 speakers. So Dr. N.B. Majumdar, shall we now open the floor for questions and answers? Is that all right?
I think now we can have the open session, and we have some questions also, not many, but we can try to find them out. And,
So I have noted down from the online that questions are coming, and perhaps we can combine when questions are common and address to the speakers, and I can see that our speakers are present here. So I'll just go from the top. So there is a very interesting question from Mr. Pankaj Jain from Sulabh International. He is asking if when herd immunity is achieved, maybe in future in a particular place, will it lower the viral indicator in the sewage? Is there any correlation that could be established or that could be foreseen? It's open to any speakers who would like to comment on this.
I can give it a first go.
Please.
It is speculative, of course, but looking at what we would expect is that if people become immune, their response to infection with the virus will mean that the virus is not capable of multiplying to the high levels that it is doing now. So the immunity kicks in earlier and tackles the virus. So my guess would be that we will observe lower virus shedding and hence also lower virus circulation in the community, hence also lower virus concentrations in wastewaters.
Actually, hard immunity is the advantage because this will, then people will be not shedding. I mean, that
Yeah.
is correct. Yes.
Yeah, that is very helpful. I think it gives perspective to how things might evolve in future. I will combine a couple of questions, particularly related to wastewater treatment plants. So some of the attendees, they have raised questions about sewage treatment plants and related aspects of SARS-CoV-2 detection. So some people are asking, how safe are these treatment plants? Obviously, Professor David Jones has mentioned the case for UK and compared with Turkey. So I would request him to respond on these. And also from the perspective of treatment, some people are asking if these sewage treatment plants are actually removing all these viruses. So these are quite mixed concerns people are raising from Terry University in India, from Philippines, and also Dr. A.K. Datta from India.
Okay, maybe I'll go first. All the evidence that we've looked at and that other people have shown is that the levels in effluent, if you have primary and secondary and if possible tertiary treatment, the levels of virus in the effluent compared to the influent are about 1,000 times lower. Clearly, for every step that you go through in the wastewater treatment plant, you have virus removal. So the virus is clearly not going to multiply during that process, it's only going to decline. So we are looking in the UK at about 1,000-fold reduction. In terms of the risk to sewage workers, we were expecting to see that, and the water companies were very worried in the initial stages that people would be sick, particularly those who are working in the sewage. Of course, the sewage workers in the UK have PPE, so they have face masks and they have protection equipment. So in essence, you know, as recommended by the WHO, if sanitary workers have the correct PPE, there is no increased risk of catching COVID-19, and all the evidence suggests that. Where people have caught in the previous SARS-CoV-1 outbreak, there's the classic Amoy Gardens outbreak, which was associated with sewage, but that was not associated with direct contact with the feces in the sewage system itself. It was associated with the aerosolization of that sewage, which is a very different matter. And I think people need to be aware that, you know, people are making lots of comparisons between SARS-CoV-1 and SARS-CoV-2, and that is not valid, really. I mean, they're very different viruses in the sense that the amount of fecal shedding is different between the two, and that Amoy Gardens is a very rare example, and I don't think it should be taken as being a commonplace.
Okay, thank you. I think it answers many people, particularly those who are asking that what treatment.
Add here one.
Yes, please.
Yeah. Issue is this, that a sewage treatment plant, if we look into, the sewage is coming, conveyance time in the city maybe 5, 6, 7 hours. From residential house going to the sewage treatment plant, it takes time. Then if we look into the unit operation treatment, now if I say with respect to the fecal coliform, say 10 to the power 7 per 100 ml, if the fecal coliform count is there in the raw sewage, after treatment in conventional, we are getting roughly about 10 to the power 3 or 10 to the power 4 per 100 ml. then we are doing disinfection. So disinfection need to be done as per Indian rule now, regulation. And new regulation says that fecal coliform in the outlet should not be more than 230 per 100 ml. So therefore, it is not 0, but 230 per 100 ml. So this is not the stringent regulation that has come. And the second one is that due to COVID-19, a direction has been given to the sewage treatment plant workers that they must use protective gears when the aeration tanks are functioning, aerators are running, there aerosol in the vicinity. So therefore, the operators may be exposed to aerosol. So therefore, now it has been directed that they must take the proper protective measures so that there is no risk associated with respect to the sewage plant operator. So occupational health hazard need to be addressed. Okay.
Yeah, my 2 cents to this issue will be that, yeah, I fully agree with Professor Jones, and because with every treatment, and in fact, I would like to add that the calculation of ours shows that the even second showing magnificent reduction, and even chlorination has a sort of equivalent removal capacity of SARS-CoV-2 than the membrane one. So actually, while the some of the contaminants or some of the things shows various at the like tertiary treatment or very advanced treatment, but here because of the having the enveloped nature of the virus, it is even chlorination just little bit supplemented by the ozonation can show up to the 3 log removal. And so it means that, and also about the aerosol, as far as my experience says, because you know that when I go for analyzing, I have to concentrate it very, very high, which means these genes are really very dilute in the water. And so in that way also, I feel that little bit, so I would go with Professor Jones that if right PP is there, there is not much things to get scared of. However, we cannot write off the infectivity. Like I showed that even I removed this wastewater story. Our paper that talks about after effluent. After effluent, if still something, and so that's why we should not write off it, but we should not get scared of it also.
So let me reiterate a couple of points people are still raising, and they are quite interrelated. Number one is, what is the viability of the virus after the treatment when it's detected in the effluent? And then others are asking, if you are trying to reuse in many cases the treated wastewater effluent, what are the consequences, for example, reuse in agriculture? So basically these questions are very related. And one question is also about how long the viruses do survive, and when wastewater enters into environmental cycle, water cycle, and goes to drinking water, what is its life cycle? So I can see they are all interrelated. So I'll be grateful if one of you could pick it up.
I think Professor Medema can start with, because he is one of the most experienced people. And if something will be left, we can add our 2 points. But of course,
I think the first thing to state then that that is a reiteration of what Professor David Jones already said, is that there is no epidemiological signals that the water cycle is a significant route of transmission. So that nowhere in the countries that have been suffering from the pandemic, there has been epidemiological links to between people and water, not in sewage workers, and so of course that's so. I think it's important to state that, and that the virus is very capable of transmitting itself the air droplet route via a close personal contact. Okay, then looking at the information that we have on the virus in wastewater or already in stool, is that it is difficult to find infectious viruses in stools and to culture infectious viruses in stool samples already. So there are reports that already the stool is not a is an environment that is not very helpful for the virus survival. So then, looking into the wastewater itself, there have been a couple of people that have been looking with in cell culture methods into wastewater. I know there's reports from Italy, from Belgium. I've seen from and I know our National Institute is looking into it, and from Turkey. Most did not find infectious virus in the wastewater, and we have to see what if the reports from Turkey are reproduced in other places. I would imagine that looking at what we know of the virus, that if there is the fecal wastes that are very fresh, so to say, that it would be possible to find infectious virus in those wastes, but that it would very quickly, the virus, the infectivity would very quickly be lost. So putting all these elements together, I think we can still conclude or estimate that wastewater is not particularly at-risk transmission route for this virus, and that a normal personal protection, as Professor Jones already alluded to, is capable of protecting us and sewage workers against well, all the viruses and many other viruses that are more infectious via water than this SARS coronavirus.
Thank you very much.
Regards, well, maybe the addition with regards to drinking water, of course, it depends on where the drinking water is harvested from and how well protected that is. But we know a lot about how drinking water is protected and how it can be at risk. And from the regulatory, the normal monitoring of fecal indicator bacteria, we can already get a good indication whether the water is well protected or is not well protected against fecal pathogens. And if it's not, I think it's at risk, but maybe more for other viruses than this SARS-CoV-2.
Thank you. I think it addresses many questions. It's very useful.
Just one point I would like to add to the professor's comment is that even because it is a enveloped virus, even its adsorption is around 4 times higher than the non-enveloped virus. So even if it is more likely to get adsorbed and sink in. So that is also one of the things that goes in favor of like less vulnerability.
Thank you. I will move discussion now to methodologies. Some questions are particularly on the use of PCR. So questions are there any other methods? Is there any other way to correlate or detect the RNA other than PCR? I remember that Professor David Jones mentioned about moving to this digital PCRs, but is it the limit that we have to stick with PCRs only?
I mean, I can just make a quick comment, really. I mean, there's the LAMP system, which people have used. There's quite a few papers published on that now, which is meant to have high sensitivity, rapid throughput within hours. We haven't used it. The reason we're switching to digital droplet PCR is that it has much better recovery at low levels. It requires quite an investment in comparison to qPCR. It's about $75,000 for a machine. QIAGEN are selling one which is relatively cheap at the moment if people are interested in investing, but it doesn't do one-step PCR. So yeah, that's as far as I know. I haven't tested the LAMP system, but other people, I don't know whether anybody else has experience of that system.
Just following on that, Manish, if you can also consider if other people are asking about any, let's say, future methods, more cost-effective methods, on-site methods, even if they're not as accurate as you want it to be. If they're
we have to understand that when people ask this question, they are asking from the sampling to analysis portion.
Yes.
So when it is purification part or enrichment part, we already have, we have told that there is a variety of things that you can go for a concentration method, and even that concentration method can be membrane based, can be several based. So concentration have the different aspects. Another thing is that we can precipitate the viruses using PEG and the NaCl. So these two are the most commonly used. So it means that then there is another method that, so we have to go for is that, of course, but scientific, and then there is a RNA replication stage, RNA extraction stage. So there also we have the several the methods to opt on, or several tweaking is possible. So it means the main thing I would say apart sampling is that the first that whether we are going to enrich the sample by filtration or we are going to precipitate the arsenic, the virus, and then the RNA extraction, that what we will do. And third, but terminology or final thing will be RT-PCR. So that is what we, probably Professor Jones also say, and that's why we don't trust, or I have not seen any paper. He's modest that he doesn't know, but we have not come across any paper where they have not end up with finally amplifying the gene and see that with the particular gene. So RT-PCR has no substitution here. We can do the substitution in these steps. We have the choices to make here. As far as some question was there about the detection label. So sometimes it is even few number of copies. Even if I would be correct, even 7 copies, 10 copies can be determined. So it is that is also, and sometimes it is semi-quantitative, because if a kit has given provided to you with some CT number, it corresponds to certain copies, you always can back calculate because there will be a CT number, and that you can have. So then there is a semi-quantitative method also. There is a quantitative method also. So there is a wide range of things. The point is that the lot of person is among those who wants to jump into the pool, and they are thinking that, okay, what will be the easiest way. But I can say is that once you will start, the things will fall place. It is not, there are semi-quantitative, quantitative, and several aspects of the things. That's, I don't know if it is.
Okay.
Well, I think now, Dr. Devendra, maybe we can hear from Asian Development Bank, and then whatever questions are remaining, the participants can write to us, and we will get the answers from the respective speakers also if necessary. Because now we have about 20 minutes left.
Yes, yes,
so
indeed.
Asian Development Bank we can hear.
Yeah, so most of the questions are covered, but broadly, but of course participants are most welcome to get in touch with us, and we can follow from there. So I think this is a good timing to invite, I think Mr. Tom Panella is here representing the Asian Development Bank team because he's Chief of Water Sector Group. Tom, are you around here?
Yes, he is there.
Yeah,
okay.
I'm here. Thank you very much. And it's a pleasure to be here. I do have a short presentation, but I think in essence of time, we can dispense with that. And I was given the task of really reacting to what I've heard in the presentations and the discussion, which is in some ways an easy assignment, but also a difficult assignment. Let me just give you a few introductory remarks on ADB. We're a development bank for Asia and the Pacific. We've been actively engaged with the COVID situation, and to date have, since the crisis started, processed about $16 billion in emergency assistance with co-financing in 41 countries. Much of this is for fiscal stimulus, as well as emergency health and other activities. But we've also been really looking at our water sector operations and how we can respond. Water is $2.5 to $3 billion a year for us, and about half of that is for water supply sanitation. So it's not an insignificant sum of money, but there are competing needs for all these investments. And so certainly detection of the virus in water and wastewater has been something coming to us from our clients as a demand for more information mainly. We've been working with KWR and some of the other people and have our own webinars on this. But I think what's clear and is that sewage surveillance is an epidemiological tool to deal with this. But then let's look at how you go about implementing it in terms of all of the issues raised, sampling, so on and so forth. And in terms of a situation of very limited public resources, how do you make the right decision on how much to invest? You know, we have some initial costs that you're looking at $1 million to $2 million to get a lab and perhaps testing protocols up in a city of 3 to 5 million people. Now, I think that's important, and I wouldn't just be talking about the SARS-CoV-2 here, but I think it's more important to look at it broader from all pathogens that can be detected. And I think Dr. Jones pointed out the difficulty in how do you sample, how do you do these things. And one, getting the science right, getting the protocols right, and then I think more importantly, who's responsible for doing this? That question was asked. And I know this isn't the topic of this seminar, but really you have Department of Public Health, you have sanitary engineering, you have the various water supply agencies. And then assuming you could test throughout the network, and I think the data shows that if you could test appropriately, it is a good indicator and can even provide lead time on when you can expect the peak, and ideally, if done thoroughly enough, where you can expect that peak. I mean, I think all of that's been demonstrated. But again, even at the most advanced centers doing this, it's not part and parcel of water supply and sanitation at this point. So I think some of the other facts that stand out is one is in the case of India, 60% to 70% of wastewater is untreated, and then from the talk that if you're going to a secondary or even tertiary treatment, you're reducing by 1,000 times, I think is what Dr. Jones said, the incidence of the virus in that. So, just by investing in wastewater, which needs to be done for many reasons anyway, you are going to be treating that virus, but I think more importantly, you're going to be treating a lot of other waterborne diseases, which kill between 3 and 4 million people every year, mainly children. So, well, I think it's very important for us to focus on this COVID situation as a means to generate this type of research. And then I think more importantly, how do we mainstream it into development of water supply and wastewater treatment systems? This broader context of waterborne disease is perhaps more important. And this is where I think really this presents an opportunity for all of us who are interested in water and in improving health outcomes across the world, that we need to take this unfortunate crisis, address the crisis, but also use it to address the broader issue of water supply, sanitation, and adequate sanitation across, in my concern, Asia Pacific, and scaling those efforts significantly. And we will continue, but I think the other points that are raised is it's also good that if you can't afford individual testing, this perhaps provides a broader way to look for hotspots. Also, you can focus it in key transmission routes, cruise ships, airports, whatever. So I think there is a lot to be said for doing this, but I think from a policy-making perspective, you need to sit back and look at what are the demands for health and water supply and sanitation in general. Then where do you place those investments where you're going to have the greatest impact? And at the same time, use the science, the public awareness, and everything that is coming out of this crisis to address the crisis, but again, more broadly address the issue of WASH and health, and giving people adequate access to water supply and sanitation, which will not only address the COVID in the short term, but have a much longer lasting impact for the world's population if we can do those things. And I'll leave it at that.
Interesting reflection. Obviously, long-term strategy is what probably is more value for all the efforts that people are going to make. When we plan this webinar, our idea was to take the ideas from best researchers around the world, bring it to communities in countries like India, Asian countries, or even Asia Pacific, and see how we can make those strategies which are basically much more impactful. And obviously, as you mentioned, there are from policy point of view, all the bodies they are interested in making investment which may be limited, but to get best out of it, addressing the health, public health, WASH and other issues in a broader context during the pandemic and after that. And that's why we are trying to synthesize everything so that we come up with ideas in many developing countries where with limited resources we can do many things as much as possible. So the host organization, IASPH, and the chairman, Dr. Majumdar, who is here, is going to take all these actions and going to discuss with his stakeholders. And of course, we are looking into India, but not limited to India. Our participants are from many other countries which have very similar situation as India, as you were mentioning, where sanitization coverage is still quite low in most of the areas. So we'll talk to the stakeholders, and we will see that how we can bring the research to advance the research in developing countries in this area, but also to advise or recommend the policy and stakeholders and other institutions so that they can make significant actions on ground as well in months and years to come. So Dr. Majumdar,
I have a little bit.
Yes, please go ahead.
Add to this because, I mean, we are doing our science, and our work is returned in terms of our publication and of course the people. But at the end of the day, we also that it has to be reflected on the field and people get benefited. And there, what I have seen is that the organization like UNICEF or ADB or WHO can play much better or effective role than the government organizations, because the government organization has, I would not say that they want to be like that, but in India, at least they have a protocol to follow. And if that protocol, a system is such that it takes time. And so there, even if they try to have a number of projects they invited on the COVID, but still I am hearing about that, okay, the review is going, because there are several points where there is not. So in my case, I would say that I have published that we could detect it in January. I have done it by my end. But what will happen is that my own institution had to fund, because initially I can put my own grant or something like that. But then institute had, and now UNICEF and WHO, which I was not much hopeful from them. But when I sent them, they are considering it very fast, and probably within this week, they should sanction it or whatever. But so in that case, ADB and this organization have the much better role. This is the time when these people who can understand and grasp and don't have much system to follow. A system means like a very long chain of the system. And so that is the one part. The second part is that I wrote that willingness, and there will be several aspects of the wastewater surveillance. Like somebody, Professor Majumdar always say 9 to 11 or 7 to 11 sampling. I don't care about the 7 to 11, because if I sample every day or 3 days or weekly 3 p.m. and every day I go for 3 p.m., 9 to 7 or 7 to 11 doesn't make sense to me. So it means, because there are several processes going on. I told you that adsorption process, even the water and the sludge component will be also taken into account. But even if all those kept constant, still we can get meaningful information. So that is one point that I want Dr. Majumdar to get it. That's why I am saying is that even if people will tell you that there are so many variables and all that stuff, you say this variable will act every day or not. If it will act every day, then it becomes a constant. And whatever the variable that I will get is the variation. So in that case, this is very much the second. And the last, but not the least, is that, of course, while making a passionate information about this, what information we'll get. And I just wanted to add that the third component is, if we can train, because several researchers, we have to invest it for future, not only for, if somebody will say, let's suppose we give a fund, we establish a lab, and suddenly COVID-19 will go, no problem. Because it can help you, because I shown you the paper 2013, where they found that coronavirus was there and respiratory. So it means it is not going to be a system for now. It is going to be a system for centuries. So that is why that is also, so just 3 points I wanted to add.
Thank you.
Can I pick up on that last point, because it touches upon something that I've been finding remarkable about the sewage surveillance that we are doing. That we've been working already for a long time on molecular methods or all kinds of methods for water quality testing. And I think Dr. Paella was also referring to how we can ensure that we can supply safe water and sanitation for all. And that, we are looking with WHO and UNICEF, for instance, for rapid methods for E. coli testing, and we're seeing that also their PCR is one of the types of methods that can serve a purpose. At least in our country, we have moved to RT-PCR also for E. coli, for drinking water testing and for bathing water testing. But the point I was going to make is that if this pandemic is raising investments into the developing countries that would support things like molecular methods to be implemented on a much wider scale, it would really empower the people and the governments of these countries, because now you can really move from the good old E. coli testing to all kinds of virus testings, the rapid E. coli testing, and when there's a new virus coming or a new parasite coming, look how it was possible to quickly adapt the method, in this case, to this new virus. So our traditional water methods, they took long times to produce a result and long times to develop. Now with this pandemic, we see that how quickly it can work with these molecular methods. So I would suggest that an investment in molecular methods for SARS-CoV-2 would have all kinds of collateral benefits in other areas. So that was the point I wanted to make as a response also to Dr. Kumar's or Professor Kumar's comment.
I think Mr. Faecal wants to say something.
Yeah, no, thank you very much. And again, I think we're all in agreement here. I think that, you know, this unfortunate situation stimulating all the science, as well as just the general awareness in water supply, sanitation, and WASH is something good. And I do know the work that KWR is doing, and we're also working with clients to try and get these advanced methods into our projects. And that's an important thing where if somebody's investing in wastewater for the first time, why not use the state of the art and what's out there, which will be quicker and in most cases cheaper. And so I think that's an area where we're very keen to collaborate with our clients, which are governments, but also we rely heavily on the input from the people like yourselves who are just advancing these methods, but we also need to get the awareness with policymakers as well. Thank you.
Yeah, thank you. So, Dr. Devendra, we are at the fag end of the event.
Yes.
So,
yes. Yeah.
Maybe I would like to say a couple of words here. You see, we have got a glimpse of the huge body of research and knowledge which is building up in different countries. And I think if we look back at what we had in the last 2 hours, so on the one hand, we have this, this knowledge about the research. The second is for developing countries where so far not much work has happened. We need to develop our own protocol with assistance, help, and cooperation of those who have worked more. And thirdly, our government, the multilateral agencies and the bilateral agencies, as Professor Manish also mentioned, so all have to come together. So forming a protocol which is acceptable to most of the people, and then to see how these things can be funded. So, pilots can be developed in different countries, in different cities, and then they can be gradually moved forward meaningfully to see how they can actually serve as an indicator. Someone said, I think a couple of the speakers said that it's time now that apart from the human testing, the environmental testing is also becoming very, very important, because I think Professor Manish again said that these are going to be there for centuries. Why centuries? These are going to be forever. You see, we have to have different kinds of microbes. Some would be pathogen for us, some would be friendly for us. But we have to prepare ourselves for living with them, fighting with them. So I think it's very important that from all the three angles, we have to come to a point we prepare ourselves meaningfully to take on this fight. Thank you. Dr. Devendra.
Thank you, Dr. Majumdar. Yeah, I would say you have very well summarized the potential action direction. That's what we wanted towards the end, that where we should be leading and making use of all these knowledge that has been developed in different places. I would say thank you to everybody, the 4 speakers, ADB represented by Mr. Tom Panella, and all the attendees, and we had many attendees from many different countries, and they managed to attend through these huge time differences. I know some people had to wake up very early in the morning, while there are other people in Manila, they're still in their offices. But we managed to gather everybody because we wanted to go in a very well thought direction from from fundamental research point of view as well as application point of view. And that's why the International Academy that Dr. Majumdar is leading, who is hosting this webinar in association with my University of Surrey, and NBH Global Limited. Dr. Hana Sinkori is also online. We came together to formulate, synthesize everything, and we thought that we can start from New Delhi, something, and definitely this can roll out to other places in India and in other Asian countries. So that is what we had in our mind. We will be in touch with all the speakers as well as participants, and thank you for those who have raised the questions. We have monitored down the questions and they are all on record. We will upload most of these materials on Academy's website, and we'll be in touch with you with links where are those information uploaded, and whatever discussions we will be having with the stakeholders, we will attempt to keep everybody posted, and hopefully this is just the beginning of a longer lasting collaboration in this area. Okay, if there are no other comments, Dr. Majumdar, then we can close this webinar at this stage.
Yes. Thank you very much, all of you. And we'll carry it forward, as Dr. Devendra said. This is just the beginning.
And we also want to thank Devendra for making all this effort and conducting.
No worries. My pleasure.
For,
thank you.
Putting me with the stalwarts like Medema and Jones. Thank you very much.
Excellent. Thank
you
you all, and
having
have a
me.
good.
Thank you very much.
Bye. Bye.
Bye.